comment on that? You know, is it like a happy, I don't even know. You know, it shows like a honeymoon for a couple who are celebrating their marriage in Italy or, I don't know, Greece or something like that. And then the story begins. I talk about that. Those of you who are interested, you go and look. But the point is that a person having multi-drug resistant TB, which is on the government watch list not to travel, and he travels with his future wife, marries, gets married in Greece, comes back, my goodness, and travels thousands of miles, and I'm talking about this news, this is public news. And the worst part is that that young girl happens to be the daughter of the guy of the CDC who is in charge of the multi-TB drug resistant department. So you never know what's happening to the person next to you. And purposely, and I tell you a story that the other thing that they learned was that not to create a panic, that's for their financial interests. So there are a lot of things that are there. There's a medicine being practiced for simple poor people, for masses, and there's something for other people. Unfortunately, that's what I think Dr. What's her name? Tatiana talks about that, about the issue of the health issue of not having insurance and so on. And there are ignorance as well, and there is, I mean, a lot of things that are happening, but keep that in mind that this is there. So anyway, water is important, post-sewage treatment, and then again, the vaccines are important. That's another, I mean, I have to teach you, whether you are anti-vaccine or whatever, but as a physician and as a immunologist, I have to teach you, and I'm going to teach you again, vaccines first. Okay. Now, I may have some other issues that you will hear me talking about in terms, and you heard me, I talk about Africa and, you know, the copyright and stuff. By the end of the day, as far as we are concerned, if you're going to ask me, and people did ask me COVID mentioned as well, I did fully get vaccinated, and I am all for people to get whatever is out there. And this will be very challenging for you, and you better read word to word 36, 37, 38, because you're going to be in consultation with the people, because Walgreens or whatever these companies are pushing their pharmacists to sell their vaccine. Students tell me that they get a bonus. So they're out there, so they have to convince the people, right? They have to sell their shingles vaccine, they have to sell their COVID vaccine, and make sure that you do a ethically, morally correct kind of information. So that's where, as I said, that I would not accept an older understanding because things have changed, and this new textbook is talking about that. Okay? All right, so let's move. And you can read all these details, you know, and then again, I'm not going to put, I can just give you an example. The most important thing that you can see from here is that viruses have symmetry. So this is symmetry, right? They are like crystals. And they, not only a crystal, but they have these receptors. You can see they have receptors, they attach to the receptors, right? And this is what I'm going to talk about today, and this is your take, and this is the pharmacist part, and I do want to spend like 20 minutes, and this is something that you have to draw, memorize at all time, and this is like, I don't know, this is like your insurance card, you have to keep it in the car. Okay, so let's begin. How do I shrink? I don't know. I shrink this and I lose it. Okay, this one go. Keep it right here. Okay. What do you see? Okay, let's go in the same order. Okay, now I expect you, and I kind of somebody sent me a note that send me a note, one of you probably, don't assume that we are preparing lectures ahead of time, don't assume that we listen to your partner, so there are a lot of assumptions, and keep on having good faith in you, but by the end of the day, you have to do what you need to do, okay? Pharmacy, antiviral therapy, okay, and you cannot graduate without that, they're gonna haunt you. So you heard so far that I talked about the viruses coming with this, attached with the cell, using take over cell machinery, divide, release, you heard me saying. So this is a typical cycle. And you also heard me saying that it's a viral, flu, cold, just take rest, symptomatic treatment, a lot of fluids, this and that. You know, and it's also in my YouTube as well. I used to drive when I was working in Loyola Medical Center. There's a big billboard. It's from Nissan company. It said that plan your sick days wisely. Plan your sick days wisely, because at that time everybody had to take sick days. I've got cold, so you can't do it every other month, but at least every 6 months. So you plan ahead of time. Whatever vacation you want to do, people will accept that. But now the question is that it's a serious issue. And I may not have time, but to tell you at a higher level of what's going on currently, maybe if you take elective with me in your P3, P4, then we can talk about all these things. For example, you say, so where is the COVID-19 right now? What's the difference between COVID-19 and flu? What is happening? You know, all these things, but again, you have to do the basic part, okay? So keep that in mind, that this is, I think I did find that. Oh no, why did I do that? So, and if you, once I give you my study guide for the finals as well, you'll see that this is something that I talk about a lot, right? So virus is out there. This is a cell, bilipid membrane. Okay. Maybe one of you guys can kind of help me. You can put it in one, right? In like one screenshot, as you are seeing over here. I don't know why I don't like display on this. Just exactly I wanted to see. Chapter 36.
What page number?
36, go for 36, and you see the chapter 36. Keep moving up, 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 up. Yeah, this one, yeah. I just wanted to kind of, yeah, that's fine, okay, great. All right. So drugs and important. Now, you can see from here, there are 9 steps. 9 steps. And I'm just talking about the general antiviral therapy, 9 steps. And I'm just kind of throwing at you. I hope nobody fails in the final exam, but if in the final exam, if you fail and come and talk to me and say, oh, Dr. Fadal, I did study biology, so I'm going to ask you to draw this in front of me, like over there. If you're able to draw that, I'm going to give you a passing grade. So this is so important a point that everything is based upon that. So 9 steps of viral multiplication. So virus number 1 comes and recognizes his favorite tissue, tropism. Hey, I'm a hepatitis virus, I found the liver, I found the lung. So this first step. Okay, I'm going to go. Second step, it attaches with it by the receptors. It has to firmly attach, right? After attachment, it has to penetrate. It has to go in. Number 3. Number 4 is going to take the clothes off, uncoating. It's going to take things off because it's going to say, you know, make this. It's going to throw it up to the cell machinery to take everything off for manufacturing, uncoated, right? And number 5, they will have proteins transcription, same biochemistry, protein transcription. When you see protein synthesis, you call it transcription, right? And then after 5, where's the 6th one? Yeah, then the protein synthesis will take place, right? Number 7, replication. Number 8, put the things together, assembly. And number 9, break apart the cell and release. This is the life cycle of a virus, of every virus. So if you go and look at the DNA viruses, RNA virus separately, you know, you'll see that the virus structure will be different, the receptor will be different, right? And DNA, RNA will have a different, something will go in the protein, something will be made in the cytoplasm, or something will be made in nucleus, right? The proteins will be different, and then cell will lyse and release. So these are the different things that will happen. And the tissue may be, this is a generic cell, but it could be any cell in the body, all the way from brain and so on and so forth. Okay? And all these are described in details in the book that I would want you to read. Right? For example, I'm going to talk about the structure of the structure of the virus. What is the different, right? And then there are receptors. This is called pathophysiology. So receptors have to bind tightly. And then if you look at the first chapter of physiology, when we talk about diffusion, when we talk of receptor-mediated phagocytosis, when we talk about how this is taken in, right? There's a process in cell biology. So that is happening. And then again, uncoating, and then so on and so forth. Now, keep that in mind. Let's take a generic virus. So if you take a generic virus, for example, you can see over here, once it comes from the attachment part, it picks up a portion of the cell membrane. That's why you see this green color structure. And then it kind of opens it up, and then there is a protein there, and then there is a DNA or RNA depending upon, you know, what the virus has, because DNA replication and RNA replication will be different. And then the proteins will be made, like all the different components, assembled together, form the exact virus that we had, and then it's going to break it apart and release. Sometimes the virus doesn't break the cell, right? And it just drifts out. Right? It has a chance, it can kind of protrude out and kind of, you know, like potato budding, like bud out, without lysing the cell, but sometimes it does and release, but the release does take place. Okay. Now, as a pharmacy, right, we are talking about drugs. So we are not talking about the measures that you already have in before, you know, the healthcare, wash hand, blah, blah, blah. We are not talking about that. Masking, you know, we are talking about treatment. We are talking about drugs. Okay, so our drug story starts from number 1. We want to make sure that we stop that attachment. We don't want the virus to bind to the cell. And there are proteins over there. We want to block that. Right? For blocking, we use antibodies. So that's the number 1 drug. I'll talk about that antibodies in immunology class in fall. As you can see, these are called antibody receptor antagonists. So if you kind of cover this up, block that, either on the virus or on the cell, so that they don't come together. If they don't come together, you stop, it says stop the evil in the bud. You stop that, that would be the best thing that could happen. Right? But that has to be has to happen very early.
So you're saying it would be best if the antagonist doesn't attach, right?
No, the virus doesn't attack.
The virus attached to the receptor, right?
Does not attach. Can you think of any drug that we have right now that could do that? Yeah, Edwina? Any drug? Yeah, Livia?
Penicillin?
No, no. You came late because we talked about viruses. Penicillin is antibacterial. So you were like last week. Yeah.
Like an antiviral medication?
What?
Acyclovir,
Say it again.
Tamiflu.
Vaccine. Vaccine. Vaccines do that. So you have a circulating vaccine in your body, having antibodies. The moment virus comes, it will not allow that to happen. That's the vaccine part.
Right, but this picture is showing if the virus does attach, right?
No, this is a complete picture.
Yeah, is that the virus though?
This is a virus. This is what normally happens and gives you the level of drugs that we have. It gives you a very generic. Like for example, it says antibody receptor antagonist.
Right.
So this is one of the things that we do is like vaccines or any drugs which are there. But the problem is in this case is how would we know that you've been infected with the virus and it's attaching? So it's like a period is unknown. So we have I mean, for example, all of us can be exposed to virus right now. So do we know that? We don't know. Unless we already have a system. We only know when it causes disease. Okay? So that's over here. So the other important thing is that we can stop this, we can stop this penetration, we can stop this attachment, or we can stop this uncoating. Now, these are the drugs which are available. Amantadine and imetadine, or all these drugs are there at level. They will stop that uncoating. So they will kind of, don't take their things off. Don't kind of send it to the manufacturing. Okay? And then again, rest is mostly protein synthesis blockers. Protein synthesis blocker. You can block the transcription, you can block the synthesis, you can block the replication. So this is a typical protein synthesis. So you have to know the transcription, translation, and protein synthesis. So that type of drugs are there which can be anti-cancer as well, because cancer does the same, antiviral as well, right? And maybe to some extent we can go for some antibacterial as well. But this is happening within the cell. Okay? And keep that in mind, if the proteins have been made, so this means there are enzymes there which are making proteins. You can block those enzymes. Right? So this is like a ocean over there. You're doing fishing in the ocean. So this, and then again in the end, you can put like an enzyme which is called protease inhibitors to stop the assembly. So okay, it has made, but we won't let it assemble or stop the release. What you're talking about is like some of the other targets that we have in terms of, Tamiflu, and this is like neuraminidase. So this is something that happens over here when it forms the receptors, so they want to block the receptors. So you can see in a generic picture over here is that whatever, and sometimes one drug works better than another. Yeah.
So like for example, like a vaccine would attach to the receptor instead of letting
The vaccines, most of the time when we say vaccines, these are antibodies.
Right.
But antibodies act outside the cell. So they don't really go inside the cell. Make sense? Yeah. Get it? So you can see this is it's a very generic thing, but it gives you an idea as to, for example, you can see from here that the virus, when it goes to budding and release, right, it picks up the cell membrane, the biolipid cell membrane of the host. Okay, can't make it. So you can see this is a host thing. So what I'm going to give you an example as well as you read the whole chapter is that it is a very generic general scheme of viral replication. If this virus is infecting like birds, right, and the cells of the birds, it is going to pick up a receptor of the bird. Right? Now, if this receptor, which is a bird, like a bat virus, it should be transmitted to the bat. But what happens is, during the process of mutation, there's a mutation. When they mutate, it becomes dangerous because it can mutate to infect human as well. So anything which is out there in birds, animals, can mutate and infect us as well. Does that make sense? So you can see this is another example of the same concept, that the attachment and penetration comes with attachment, and then again some of the viruses, the structures are such that they need to enter into the nucleus. You can see this virus has to go into the nucleus. It has to go into the nucleus because it is as a DNA genome. Because you have to understand the protein biochemistry as well. What is exactly, happening in terms of the structural part, because it is containing a message. So message is in DNA or RNA, right? So this message has to be communicated to the nucleus. So it has to go to the nucleus, and the nucleus has to then send a messenger RNA, because it's a DNA virus. And then it will synthesize the typical protein synthesis in terms of Golgi apparatus, in terms of ribosomes, in terms of structural proteins, assembly, and release. So this is more of a protein biochemistry in a way that is happening in terms of viruses, because the proteins are important. So we can have all protein inhibition or protein synthesis blockers in this case. You can see from here. Okay. And it's giving you many examples over here, and I think what I would do is that if you pay attention to it's going to give you all the details of what is going to happen in assembly, what's going to happen in release. So this is more of a little bit of genetics, a little bit of protein biochemistry, and you have to know the difference between the translation, transcription, DNA, how the DNA is broken up, and DNA has to go into the nucleus as compared to RNA, which already has a sequence. So a little bit of, as I said, basic genetics for biology. Any other questions? So we've got 5 minutes, so I'm going to take questions from you. And then, as I said, that I would want you to 36, 37, 38, read it on your own, and then also do the log assignments. And keep that in mind, these Elsevier case studies are from the book. They're not from my lectures. My lectures are old. My lectures are giving you the basis of, it's more of a foundational thing, right? But if you want to go a little bit in detail, I mean, that's enough, but not enough to answer those questions, because they are more detailed. And then again, since they're open timed, so you can kind of go and, so it kind of gives you assessment for reading. So it's more of your reading exercise. And I do talk about some of the drugs, as you will see. And I think what I intend to do is, next week, right? The week after is, I already gave it to Dr. Puglisi because it was like last week. So next, I will pick up one DNA virus and one RNA virus and discuss with you. But again, as I said, that you need to do more. There's more of a self-directed learning, more of what you call, ball is in your court, you need to kind of spend some